capacity is about 10 billion, and we're at 7.5 billion or something. We're going to get to 10 billion soon. We're exponentially increasing. And so we want to be looking at technologies that can increase our carrying capacity so that we can have more people on Earth. And one of the ways that we want to look at this is exploiting the ocean using aquaculture. And the reason why is that we haven't done that much there before. And there's a lot of open space, there's a lot of room for improvement of species, and we have done a lot of improvement on agriculture, terrestrial crops. And if we apply the lessons that we've learned for 10,000 years, along with next generation sequencing, we can speed up this process really quickly. So up here I have 3 different types of brown macroalgae that we're looking at, and they all have distinct uses potentially. So Dictyota on the left, when used as a feed supplement for cattle, can reduce the methane produced by up to 90%. And so while we're not eating Dictyota, it can still have a huge impact on the environment, and we all like beef, so we all want to keep eating beef. So that allows us to keep doing that. In the middle, giant kelp, that's what we're working with right now, and that's a biofuel application. And the reason why we're looking at using giant kelp, it grows locally, it grows really fast, and that's basically all we need. Because and that's the great thing about aquaculture, is we don't need to have that much input. It can grow using the sun and using available nutrients in the ocean, and so there's a huge room for expansion if we are able to grow it in commercially. On the right, we have a brown macroalgae, Indoria pinifida, which is used for food and is grown at a high rate in Japan. They call it wakame. So here we have a broad spectrum of possibilities for brown macroalgae uses. And so just to give a little plug on the project that we're working on now is we're trying to domesticate Macrocystis in 3 years. And so that sounds pretty daunting. But using next generation technology, so we're going to assemble a reference genome, just like Nate was talking about doing GWAS, or genome-wide association studies. We're going to do the same thing. We have a test farm planned in Catalina, and we're going to do something called the test cross, grow up 2,500 kelps, look at the ones that are best growing and worst growing, compare them genetically, do a second round of crossing, compare that genetically, and hopefully have kelp ready for market. But when we're thinking about applying our kelp towards more of a global market, we started running into some problems. And one of those problems is scale. A lot of the kelp aquaculture is grown in coastal environments where there is like natural environments for kelp. And so if you start thinking of how can we actually make an impact in the energy industry, we need to grow way more kelp than what is being grown near the coast. And so if we want to do open ocean farming, we have to figure out permits for it. And permitting, as Doug probably knows, pretty difficult to get. And one of the reasons why, especially for aquaculture, is we learned a lot of lessons from agriculture. And what did we lose in terms of agriculture? A lot of genetic diversity, a lot of natural species. And we don't want to make that same mistake for aquaculture. We want to preserve as much natural genetic diversity as we can. And so you can't really just grow kelp and make it better and then put it in the water and expect it to have no effect on natural populations. So we want to try to figure out a way to get around these problems. And one of the ways that we were thinking is to leverage a unique property of brown macroalgae. And so on the left I have the kelp life cycle. It's haplodiplontic, so it means it has a haploid and a diploid stage. And what is most unique about kelp is that the haploid stage, and this is a very poor analogy, but think of it as a seed. The seed itself can create a copy of itself in the lab in the right conditions. And so rather than having to produce new seeds for each generation, you can create a seed bank that you can propagate basically infinitely in the lab for as much as you want to expand your crop. And this means that it has huge potential for generating the exact same phenotype of kelp that we're trying to grow. And so, but how do we actually get permitting for doing things like this? Well, if we use this two-stage life cycle to make sterile kelp. And so, what I want to highlight is in this two-stage life cycle, this is what you see in the water, but this is what we could do in the lab. And in the lab, we could look at genes, because we're going to be assembling the reference genome, look at genes that are only expressed in the adult stage and identify genes that are very important, essential for reproduction. And if we either knock them out using CRISPR or find natural variants in the wild, remember, as a sporophyte, as the adult, they are diploid. And so if you have one bad copy of a sterility gene, you can still produce the next generation. But if you combine two gametophytes with mutant copies of the same sterility gene, you can produce a kelp that will not be able to produce the next generation. And so being able to use this two-stage life cycle, we think we have a good potential to greatly increase the amount of kelp aquaculture responsibly in the open ocean. And so just a quick little plug on sterile kelp. Remember, one of the biggest problems with expanding kelp production is permitting and worries and very valid concerns about invasive species and transgene flow in natural populations. And sterile kelp directly addresses these problems. Another major benefit is when you are scaling up your farms, you really need to be able to know when things are ripe, when things are ready to be harvested. And that comes from phenotypic consistency. And the way we get phenotypic consistency in agriculture is inbreeding. And that's bad for a lot of ways, but the main thing is it cuts out in your genetic diversity. So you do really well in one specific environment. You don't do well when things start changing. And so being able to have phenotypic consistency by having millions of gametophytes that you can produce the exact same kelp with, while not having to do massive amounts of inbreeding in order to get it, is a huge advantage. And last thing, when you cross two different populations, usually you see a boost of growth compared to not doing it like that. That's called heterosis. And we can have heterosis at every generation because we can be doing basically a novel cross every time before we start planting. So that's a quick dive into the kelp world, and we'll do questions.